So yesterday we got as far as describing the innervation of the lower motor neurons by the upper motor neurons. We talked about those neuronal cell bodies of the upper motor neurons located in the cerebral cortex, and in fact in a particular region of the cerebral cortex known as the precentral gyrus. That neuron had a single axon, a single process that extended all the way from the cerebral cortex all the way into the spinal cord. It crossed over in the decussation of the pyramids, went to the other side of the body, descended the cord in the lateral corticospinal tract, and then left the corticospinal tract in just the right spot that would innervate the lower motor neurons. And that tied together the idea of the somatotopy of the upper motor neurons and the innervation of the lower motor neurons, which were then going to innervate a particular series of muscles. We'll begin with that today. We'll begin with the idea of what the lower motor neurons are going to be doing. And we're going to start with an important concept when we talk about lower motor neurons innervating muscle, innervating skeletal muscle. The first part of the discussion today is going to be all skeletal muscle, and I hope by the end of the second lecture we can get to a little bit about smooth muscle. We'll see. When we start talking about lower motor neurons and skeletal muscle, there's a concept that holds that you'll need to know, and that is the concept of the motor unit. The motor unit has a definition. The motor unit is defined as the lower motor neuron and the group of muscle fibers that innervates. So it includes both the lower motor neuron and a group of muscle fibers. Now, one of the things we're going to see as we move through this is that each lower motor neuron innervates multiple muscle fibers. In some cases, maybe a dozen or two, in some cases several hundred. Small motor units will have only one motor neuron and maybe 40 or 50 muscle fibers. Large motor units may have one motor neuron and several hundred muscle fibers. And we'll talk a little bit about that. So we'll lay out this idea of the motor unit, and then we're going to think a little bit about the cellular details of how the muscle cell receives information from the nervous system. At that interface that we talked a little bit about, we talked about John Langley, that interface between the lower motor neuron and the muscle fiber, the lower motor neuron and the muscle cell. We'll see that acetylcholine is the neurotransmitter. We'll see that the receptor is a nicotinic acetylcholine receptor. And we'll see that unlike in the central nervous system where we got rid of excess neurotransmitter by pumping it back into the glial cells, here in the neuromuscular synapse, we're going to use enzymes. There's an enzyme called acetylcholinesterase. And acetylcholinesterase is going to break down the acetylcholine into choline and acetate, neither of which are capable of binding to the nicotinic acetylcholine receptor and opening it. Then we're going to talk about the skeletal muscle cell, because one of the ideas is we're going to try to talk about how we communicate between the nervous system and the muscle. And we'll see that the skeletal muscle cell has a particular membrane system to it that allows it to function. The muscle cell is a really strange cell. It's unusual in that it contains hundreds of nuclei. It's a multinucleate cell, and it arises as a multinucleate cell during development when individual myocytes, as they are developing, will fuse one to another, and each of the myocytes contributes its nucleus. So in some muscle fibers, some muscle cells, there'll be 100 to maybe 1,000 nuclei, because that's the number of myocytes, myoblasts, that had fused to make that muscle cell. But we'll see then that because of its unusual size, both its length and diameter, the unusual size of the skeletal muscle requires a fairly elaborate membrane system for communication from the outside world to the inside world. And that elaborate communication system, that elaborate membrane system, is going to be the sarcolemma, and we'll draw all this out. The sarcolemma, which is the plasma membrane. An extension of the plasma membrane called the T-tubules. Those dive deep into the muscle fiber. And then there's going to be a junction called a TSR junction, where the T-tubule comes precious close to the sarcoplasmic reticulum but doesn't quite touch it. Those two membranes are going to be linked by proteins, and we'll see those proteins now are important in conveying an action potential from the T-tubule membrane, which is really an extension of the sarcolemma, the plasma membrane, to the endoplasmic reticulum or sarcoplasmic reticulum. It's going to be a linkage which is going to allow us to release calcium from the internal storage compartments. And this is the idea of excitation-contraction coupling. And what's going to happen at excitation-contraction coupling is we're going to be propagating these electrical signals along the plasma membrane, down the T-tubule system. And the T-tubule system is going to have bridges between it and the sarcoplasmic reticulum. And those bridges are channels. One set of channels in the T-tubule will be a calcium channel called a dihydropyridine receptor. We'll see we don't need the calcium to come through the dihydropyridine receptors in this story. When we come to heart, we'll see it has to come through. But in this story, the dihydropyridine receptor is going to be mechanically linked to a calcium channel in the sarcoplasmic reticulum. And when the action potential arrives, it's going to pull the plug out of a bottle, as it were, and let calcium pour out of the sarcoplasmic reticulum. That's the initial step in excitation-contraction coupling, will be the release of calcium from the sarcoplasmic reticulum. That's all going to be explained by the organization of the membrane systems, the plasma membrane, the T-tubule, and the endoplasmic reticulum. But then in order to get contraction, in order to get shortening of the muscle, we have to have the calcium interact with the cytoskeleton. And so we have to think a little bit about the cytoskeletal organization of skeletal muscle cells. And we'll see that there's a fairly short list of molecules we have to know about. First, there's something known as the thin filament. Now, the thin filament is commonly thought of as an actin filament, and that's true. The thin filament is made of actin. It's made of G-actin polymerized into filamentous actin, pretty much the same way as we saw early on in the discussions about the cell. So we're going to have filamentous actin polymerized from G-actin. But that's not enough in this system. In addition to that, we need to have, in the thin filaments, we need to have tropomyosin and troponin. Now, I ask you to know this story by heart, and that is that the actin thin filaments are a bundle of actins. But associated with those thin filaments will be two other molecules, one called tropomyosin and the other called troponin. Troponin is a calcium binding protein. If you get down into the details of this, there are three parts to it. But the one we're interested in is called troponin C. It's called C because it binds calcium. The troponin C binds the calcium, moves the tropomyosin, and when the tropomyosin moves, it opens up binding sites in the actin so the myosin can reach up and touch it. So that brings us to the next character. These are the myosin thick filaments. So we're going to have actin thin filaments and myosin thick filaments. And the way the system is going to interact is calcium is going to be released from the internal storage compartment. Calcium will bind to troponin C. That changes the charge distribution on the troponin and tropomyosin molecules, and the tropomyosin molecule moves. When the muscle is not moving, when it's at rest, those tropomyosin molecules are occluding or covering up the myosin binding site in the actin. So the tropomyosin prevents the myosin head from reaching the actin thin filament, but when we roll that out of the way, now the binding site's open, the myosin head can reach it, and that begins something known as actomyosin bridge formation. We'll actually glom that head of the myosin head glom the head of the myosin onto the actin. And then because the myosin is an ATPase, it will have converted ATP to ADP and PI. The ADP and PI will leave, and that causes power stroke. The myosin head tugs in the actin thin filament, and we'll see that's going to draw the next character together, the Z lines. The Z lines are made of something known as alpha-actinin. Alpha-actinin is an intermediate filament protein, but it's an actin binding protein. So its major player here is going to be its bind to actin. When we put all of this together, when we put the myosin thick filaments and actin thin filaments and the alpha-actinin Z lines together, we end up with a kind of cytoskeletal organization, interrepeating pattern of these Z lines, actin thin filaments, and myosin thick filaments called the sarcomere. The sarcomere is a subcellular structure that's highly organized, and it's organized at its ends with alpha-actinin. And in between, there'll be actin thin filaments attached to the alpha-actinin and myosin thick filaments between the actin thin filaments. And we'll draw all of that out. We'll describe this. But when we think about a skeletal muscle, what we imagine is that we have the cytoskeleton organized into these sarcomeres so that The sarcomere itself is only about, a micrometer, maybe micrometer and a half long. But even a small muscle cell will be a millimeter long. So there'll be like 1,000 of these, even in a tiny muscle, lined up end to end. And then when we shorten each one of the sarcomeres by, say, 20%, we then shorten the entire muscle by 20%. And we'll walk our way through how that works and what that means. And then the next thing is to think about how we stop the process. And we're going to basically reverse the whole story. And it's going to reverse by stopping the arrival of action potentials at the axon terminal of the lower motor neuron. We'll stop releasing neurotransmitter. We stop releasing neurotransmitter, we stop generating EPSPs in the muscle fiber. We stop generating EPSPs, we no longer have action potentials. So now we're no longer propagating action potentials along the plasma membrane and down the T-tubules. So we're no longer releasing calcium from internal stores. So now what happens is the pumps take over. And the pumps start to remove the calcium from the cytoplasm. They basically pull that calcium out of the cytoplasm, stick it back into the sarcoplasmic reticulum. When they do that, the calcium levels in the cytoplasm begin to drop. They begin to fall. And as those calcium levels begin to fall, then we see the calcium comes off the troponin C. The tropomyosin molecules move back over the binding sites on actin for myosin, and the myosin can no longer reach up and touch the actin thin filament. That's how relaxation occurs. There's one more piece you'll need to know about, though. When that myosin head is attached to the actin thin filament and it undergoes its movement, it requires additional ATP to have it uncouple from the actin. So the binding of ATP is going to uncouple it from the actin and reset it. And then we can start the process over and over again, drawing the Z lines together until we stop having action potentials arrive at the axon terminal and go through the whole process, pump the calcium back in, and the myosin head will let go of the actin thin filament. That brings us to a concept that I'll discuss. It's a little gruesome, but it's the idea of rigor mortis. Rigor or rigor mortis is the stiffening of the body after death. And we can explain that through these machinations of what's happening to the calcium, what's happening to the loss of ATP, and so therefore we can't pump calcium into the sarcoplasmic reticulum. We no longer have ATP, so the myosin head can't let go of the actin thin filament, and we've frozen those muscles into this contracted state with the myosin head attached to the actin thin filament. So let's start here with the idea of the lower motor neuron. And we'll say that the lower motor neuron innervates muscle cells in a muscle, a skeletal. Now, when we talk about skeletal muscle, what we mean are the muscles that are attached to the skeleton. And we'll often refer to the skeletal muscle as voluntary muscle. And it stands to reason because we said yesterday we had those upper motor neurons and we would choose which upper motor neurons we wanted to activate. Those upper motor neurons, once activated with action potentials, would turn on lower motor neurons, which in turn turn on muscle fibers. And that's what we're going to do today. We're going to activate the muscle fibers. To think about how this is going to happen, though, there are a couple of terms we need to talk about. First is the so-called motor unit. And the motor unit has a definition. It's called the lower motor neuron and the group of muscle fibers. When you see this term fibers, when we're talking about skeletal muscle, you can read muscle cells. And the group of muscle fibers it innervates. As an extension of that idea, we can say that each lower motor neuron, LMN, innervates several muscle fibers. But having said that, each muscle fiber is innervated by only one lower motor neuron, at least in the adult. So that's basically the plan. We're going to have innervation of upper motor neurons, we're going to have innervation of lower motor neurons by upper motor neurons. The upper motor neurons are going to drive the lower motor neurons in that myelosynaptic pathway. They're going to bring the membrane potentials of the lower motor neurons to threshold, fire action potentials. The action potentials are going to propagate along the axons of the lower motor neuron, and they're now going to activate the muscle fibers. What this tells us is that each time we turn on a lower motor neuron, we're going to turn on several muscle fibers. So when you think about a muscle that has 1,000 muscle fibers in it, you may have maybe 20. You may have 20 motor neurons innervating that muscle with 1,000 muscle fibers. Each one of those motor neurons then would innervate 50 muscle fibers. So one motor neuron, multiple muscle fibers. So any questions on that? So then, with that idea, let's draw something out and see if I can illustrate this with sort of this simple drawing. And we'll start again with the cross-section of the spinal cord. The gray matter. And down here in the ventral horn, or the anterior, same thing, we're going to have the cell bodies of the lower motor neurons. One of the ideas here is that all the lower motor neurons have their cell bodies in the ventral horn, the gray matter of the spinal cord in the ventral horn. So I'm going to put one in here. I'll show it as a red one. And then over here, I'm going to draw up a series of parallel lines. And each set of these parallel lines is meant to be a muscle fiber. I'll have to number them to keep them straight here in a minute. So we can imagine that this is a muscle. This is meant to be a muscle with many muscle fibers or cells. Let's number these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Oh, let's put another one in here, 14. Now we're going to come back here to the cross-section of the spinal cord. We're going to look at lower motor neurons in one segment. I'm going to put a few of them in here. So the first one I'm going to put in is red. So this sends a process out into the periphery, goes into the ventral root, joins the spinal nerve, and finds its way to muscle. And it runs its way along the muscle during development, and it will find places to innervate. It's going to innervate 4, 7, and 9. Meanwhile, blue, during development, does the same thing. Blue sends a process out. It, however, innervates 1, 2, 6, and 8. Green, again, cell body in the ventral horn sends a process out, joins the spinal nerve, and innervates, say, 3, 5, 10, and 12. And black, another motor neuron, does the same thing, and now it innervates. I have to make sure. It's going to innervate 11, 13, and 14. So this fits the story. Each lower motor neuron innervates multiple muscle fibers, but each muscle fiber receives input from only one lower motor neuron. So that's the pattern we've drawn here. Now let's imagine what happens when one or the other of these lower motor neurons becomes active. I'll remind you that they'll receive input from the neurons in the lateral corticospinal tract. This upper motor neuron sends a process over and will innervate one of these lower motor neurons. So now when the upper motor neuron activates this lower motor neuron, let's choose black, then when it sets up EPSPs here that generate action potentials, those action potentials will propagate out into the peripheral nervous system, propagate along this set of this axon, and now find the synapse at 11, 13, and 14, causing 11, 13, and 14 to contract. So only 11, 13, and 14 are going to contract because the only lower motor neuron that's been activated is black. You can do the same thing with any of the others. You could imagine activating red, but now when we activate red, the action potential is going to propagate to the terminal here, and they're going to activate 4, 7, and 9. Now I've shown this as being very, very small motor units. Typically when we talk about small motor units, we're talking about one lower motor neuron and maybe 30, maybe 40 muscle fibers. That's a small motor unit. Where you'll find those tiny motor units is particularly in muscles that activate the hand up here in your forearm, where you can do fractionated movement of your hands. Those are very small motor units because what you can do with those fine motor units is you get very delicate control. Another place where you find really small motor units is in the extraocular eye muscles. These are the muscles that operate your eyeballs. And if you've ever watched someone read, you know you can watch their eye move across the page, move down, come back, move across the page. That's very, very fine movement. And it's such fine movement that we require very small motor units to get those very delicate and fine movements. And on the other hand, we can imagine that we have fairly small motor units in the muscles operating our hand. But we can still get a lot of grip strength with our hand. I mean, you can go over to a table and pick up a dime, and then you can move over and pick up a suitcase. And how do we manage to get this combination of dexterity and strength? Well, the strength comes from the fact that what we can do is even when we have small motor units, we can begin to recruit multiple motor units. So when you really need to generate a lot of strength, what you're doing is you're recruiting many motor units. In reality, these motor units remain on only for a short period of time, maybe a couple of seconds. Then you rotate that one out of the line, bring another one on. So that's why you can exert muscle activity for long periods of time. And much of conditioning, particularly weight conditioning, has to do with has to do with getting to the point where you can hold weights for long periods of time, as you can circulate many of these motor units on and off and on and off for long periods of time. That's really a large part of conditioning. But this is the concept of the motor unit. Let me show you a picture of this. Sort of pretty picture of what I just said. And so when we look at this, what we're seeing, here's the spinal cord, here's the ventral horn, here's the ventral root, this is the spinal nerve, and they're showing just two lower motor neurons. And they're showing the lower motor neuron in this sort of magenta color coming out and innervating these two muscle fibers, and the purple one innervating these three. But these endings here, those are the synapses, the end plates that I showed you the other day in that picture. And it's the same idea here, but each muscle fiber will receive input from only one lower motor neuron, but each motor neuron innervates multiple muscle fibers. And when that lower motor neuron becomes active, all of the muscle fibers it innervates contract at one time. That's the whole concept of the motor unit. Now the next thing is a highly stylized diagram, and this is what we're going to draw next. What we're looking at here is the kind of interface between the lower motor neuron and the muscle fiber. This is going to be the synapse. This is going to be the neuromuscular synapse. And I'm going to draw this in just a minute. And what we'll see is that when the action potential arrives at this site, it's going to release neurotransmitter through those mechanisms we just talked about earlier. That neurotransmitter is going to be released onto the postsynaptic cell. There are going to be nicotinic acetylcholine receptors on the postsynaptic cell. Those nicotinic acetylcholine receptors are typical ligand-gated ionotropic channels. Those are the ones that Langley discovered when he was using his brush with nicotine. And when we open these nicotinic acetylcholine receptors, we generate a depolarization in the muscle. But one of the things I want to impress upon you, and you can see a little bit of that in the previous slide, is that these muscle fibers are enormous. They may be 100, they may be as much as 1 or 2 or 10 centimeters long. The diameter of this synaptic site may be no more than 100 micrometers at largest. And when you actually calculate the total area of the muscle fiber that's covered by one of these synapses, it's less than 0.1%. Well, if you think about that for a moment, now the numbers aren't important, but the idea here is that such a small area of the membrane of the muscle fibers covered by the nicotinic acetylcholine receptors is that if we activate all those nicotinic acetylcholine receptors and ions come through at those sites, you can't propagate that potential very far from the synapse. So that means there has to be some level of regeneration. We're back to this idea of going from an excitatory postsynaptic potential to going to an action potential, and we'll walk our way through that. So let's do that now. So any questions up to there? So what we're going to do is we're going to first talk about the interface at this site, the synapse. We'll take this panel and we can label it as the neuromuscular synapse. And we're going to draw this out and describe what's going on at this site. And some of this will be review, because much of this is the same discussion we had earlier dealing with the snare complex. Much of it is going to be the same discussion we've had earlier with exocytosis, the opening of ligand-gated ionotropic channels on the postsynaptic side. In fact, much of that story was initially worked out at the neuromuscular junction because the neuromuscular system, the neuromuscular synapse rather, is readily accessible. So let's take this panel and talk about activating muscle at the neuromuscular synapse. We can imagine a neuron, and we can imagine a muscle fiber. This is meant to be a muscle fiber. In blue, we have one of these lower motor neurons. And it forms a synapse between the muscle fiber and the lower motor neuron. There's always a gap between the lower motor neuron and the muscle fiber membrane. This is said to be the synaptic cleft or gap. If we blow a bit of this up, if we take, say, this bit and blow it up so that we can look at individual vesicles, we find again that there's some of these vesicles that are clustered right at the membrane. These are the so-called docked vesicles. They're docked at a site called the active zone, where we also include these voltage-gated calcium channels. So up above, we've initiated an action potential in the spinal cord, in the ventral horn of the spinal cord. Those action potentials will now propagate into the periphery on those lower motor neurons, and those action potentials now will arrive at the axon terminal. When they arrive at the axon terminal, sodium influx provides positive charge. To open, ligand gated. To open calcium channels. Again, you can see it's positive charge opening, so that's a voltage gated channel. The voltage gated channel is being opened by the sodium arriving via the action potential. The calcium comes in. The calcium then binds to synaptotagmin. The synaptotagmin then instructs or permits the alpha helical regions of VAMP, also known as synaptobrevin, syntaxin, which is a transmembrane protein in the plasma membrane, and SNAP25 to all wind together and smush the vesicle into the plasma membrane. And that use of the snare complex to smush the vesicle membrane and the plasma membrane together allows for the release of neurotransmitter. The neurotransmitter here is acetylcholine. It's basically a choline molecule linked to an acetate by an ester bond. The acetylcholine is then released. And now if we look at the region here on the postsynaptic membrane, on the postsynaptic membrane, let me draw this up here in red. Thought I grabbed a red one. On the postsynaptic membrane now, stacked in really, really closely, will be these nicotinic acetylcholine receptors. The nicotinic acetylcholine receptors are atypical. In fact, they're one of the first ones discovered. These are atypical ligand-gated, ionotropic channel. The typical nicotinic acetylcholine receptor here now is going to bind the acetylcholine. We'll bind two molecules of acetylcholine, open the channel, and now allow for sodium to come in. This influx of sodium generates an excitatory postsynaptic potential, an EPSP. These EPSPs tend to be pretty big. Lots of neurotransmitters are released, typically at one of these neuromuscular synapses, something between 50 and maybe 200 synaptic vesicles fuse to the axon terminal to release their contents. Each of those vesicles contains about 5,000 molecules of neurotransmitter. So we're simultaneously opening up a lot of these nicotinic acetylcholine receptors. And if we put an electrode in here and measure this, we see the potential typically goes from minus 70. Sometimes it'll go up to minus 40 millivolts. Lots of neurotransmitter opening, lots of channels producing a sizable depolarization. Well, that minus 40 millivolts is almost always well above threshold. So we can put a dotted line in here someplace and call this threshold. And of course, if we're going to have the idea of threshold, we need to have a series of voltage-gated channels. And what we find is in this region, right nearby to the axon terminal, we're going to find voltage-gated sodium channels. They'll, of course, be accompanied by the counterparts, the voltage-gated potassium channels. So you have a kind of handoff here. You have the action potential arriving at the axon terminal. It then allows for the opening of calcium channels, allowing the calcium to come in, leading to the fusion of the synaptic vesicle with the plasma membrane, leading to the opening of these ligand-gated channels to produce an EPSP. But that's just the first step. Electrically, the generation of the excitatory postsynaptic potential is just the first step, because that influx of sodium is now going to open the voltage-gated voltage-gated sodium channels, and we're going to fire an action potential. We need an action potential. One of the reasons we need an action potential is that this muscle cell, this muscle fiber, can be as much as, maybe a centimeter long. Well, that's just too far for the electrical signals generated by the opening of the nicotinic acetylcholine receptors to travel. Just can't travel on this membrane. And we're going to see in a moment, there's a lot of membrane here. So we need to regenerate that signal. We need to regenerate the signal of the excitatory postsynaptic potential, and that regeneration is carried out by the opening of these voltage-gated sodium and potassium channels to initiate an action potential that propagates along the membrane. Basically, a reiteration of the story we've told over and over and over again. So the next thing we want to think about, though, is what becomes that action potential and how we go from generating an action potential to getting muscle contraction. So any questions up to here? Okay. The next thing I'm going to draw, I'm going to erase our motor units up here, and I'm going to draw a kind of cartoon of an individual skeletal muscle cell. So we'll take these parallel lines, and I'm going to draw what I consider to be more or less an individual skeletal muscle cell, and try to point out some of the peculiarities of these muscle fibers or muscle cells. They're very strange cells, not only because they're big, and not only because they have such a highly organized set of skeletal organization, but also because of their elaborate membrane systems. So what we want to do here is begin talking about the membrane systems of skeletal muscle cells. Take this panel, label it as the membrane systems of skeletal muscle cells. So I'm going to draw this as a giant oval. This is meant to be a muscle cell. If we look at this muscle cell, one of the first things that strikes us is that all along, I'm going to label this as the plasma membrane. Now, the plasma membrane in muscle gets another name. It's called the sarcolemma. So the sarcolemma is going to, of course, surround the cytoplasm of the muscle cell. So that's the plasma membrane. And then we see that at intervals in this plasma membrane, in fact, if you were down on the surface, you'd see every micrometer or two, there's an extension of the plasma membrane into the cell. What was I going? Sorry, lost track. The other point I wanted to make here is that when we look at this cell, we'll see hundreds of these nuclei. So each one of these blue ovals is meant to be a nucleus. So what we can say, and we'll spell it out over here, is that skeletal muscle cells are multinucleate. just means they have lots and lots of nuclei. And we know that because we know how these muscles develop. We know what's happening is that we start out with individual cells called myoblasts. During development, we have these myoblasts, and each one of them looks like a garden variety cell with a nucleus. And there are signals then between these myoblasts that tell them to fuse one to another. So this myoblast fuses with its neighbor, which fuses with its neighbor. And each one of these myoblasts contributes a nucleus. So when we count the number of nuclei here in one of these skeletal muscle cells, that tells us how many myoblasts gave rise to that muscle cell. It's not unusual to see several hundred in here. And so what we're seeing then is the muscle cell, this gigantic muscle cell, which may be as much as, 80 to 100, that would be a big one, micrometers in diameter, and maybe a centimeter long. That each of these cells contains hundreds of nuclei. And it makes a certain amount of sense because these nuclei are the genetic material. And the genetic material that is going to have to be making proteins for this enormous cell. And so we need multiple nuclei to keep up with the protein synthesis requirements of this gigantic cell. But the other characteristic that strikes us is that if we stand on the surface, imagine standing on the surface here, and walking along the surface of the muscle, we'd only walk maybe a micrometer, maybe 2 micrometers, and we'd encounter this cavernous hole. Now, if we could shrink ourselves small enough, we could theoretically rappel down this side of the wall of this pit, walk across the bottom, and climb out the other. All of that is to say that this is continuous membrane. This structure has a name. It's called the T tubule, where T stands for transverse. The transverse tubules got the name because they extended across the muscle fiber. So they're traversing the muscle fiber. But they're in fact nothing more than an extension of the plasma membrane. We know that now. And in fact, the same kinds of molecules we find in the plasma membrane, the sarcolemma, we're going to find here in the T-tubule membrane. So we can say here that the T-tubule membrane is an extension of the sarcolemma. So these are combined the first characters in the play. They're going to be the sarcolemma and the extension of the sarcolemma into the T-tubules. But if we look carefully at these cells, we'll see that the T-tubule membrane comes really, really close to this intracellular bag. Never quite touches it. And this intracellular bag has a name called the sarcoplasmic reticulum. Now, the sarcoplasmic reticulum is just a highly elaborated form of the endoplasmic reticulum. But to be sure, what we're going to use the sarcoplasmic reticulum here is the sarcoplasmic reticulum. What we're going to use it for is it's going to be a calcium storage organelle. Remember, early on, we established the idea of the endoplasmic reticulum as being a kind of plastic bag inside the cell. Well, this is just an elaboration of the endoplasmic reticulum. And that plastic bag now inside the cell is going to be filled with calcium. Now, there's this site here where the T tubule comes really close to the sarcoplasmic reticulum, and this has a name. This is the so-called TSR junction. It's just the site where the T tubule membrane approaches the sarcoplasmic reticulum membrane. They never quite fuse, they're always separate. But there are bridges. We can see bridges between the T tubule membrane and the sarcoplasmic reticulum membrane. And those bridges are composed of particular proteins. There's going to be a protein here in the T tubule. And I'll ask you to know this name, even though it seems arcane. This protein is known as the dihydropyridine receptor. The dihydropyridine receptor is often abbreviated DHP. Turns out the DHP receptor is in fact a calcium channel, but we're not worried about the calcium here. We'll worry about it when we get to the heart, but not here. The dihydropyridine receptor becomes a kind of voltage sensor. And if we look closely here, and I'll draw this a little bit larger in a moment, the dihydropyridine receptors in the T tubule are perfectly lined up with these green channels here called ryanodine receptors. Turns out the ryanodine receptors are proper calcium channels in the sarcoplasmic reticulum. And if we blow that up and look at this, what we see is that in the T tubule, there's this transmembrane protein which has a kind of foot on it that actually sticks down into the ryanodine receptor channel. So again, the dihydropyridine receptor is in the T tubule membrane. The ryanodine receptor, which is a calcium channel, is located in the sarcoplasmic reticulum. There is a matchup, a perfect matchup of the dihydropyridine receptor with the ryanodine receptor. This sort of foot that I put on here is literally blocking this channel. Another way to think about it is though this is a cork in a bottle and the ryanodine receptor is the bottle. Now remember what we have in here. We have calcium. And the calcium concentration inside the sarcoplasmic reticulum, it's a little tough to measure because it's bound, but it's probably in the neighborhood of 2 to 3 millimolar. The concentration of calcium outside the sarcoplasmic reticulum in the cytoplasm is in the neighborhood of 0.1 nanomolar. So you have a whopping driving force. And what's going to happen here is that the action potential is going to arrive in the lower motor neuron. It's going to release the neurotransmitter acetylcholine. The acetylcholine binds to the nicotinic acetylcholine receptor. That opens those channels to allow sodium to come in to generate EPSP. The generation of that EPSP starts an action potential now, which propagates from the synaptic site all along the membrane. In addition to propagating along the membrane, it propagates down along the T tubule system, delivering positive charge in this narrow gap. So sodium ions now are going to be delivered to this narrow gap between the T tubule membrane and the SR membrane. That increase in positive charge, the sodium, is a signal to this dihydropyridine receptor to pull its plug from the ryanodine receptor. It's a voltage sensor. When it sees that voltage, it pulls the stopper out of the ryanodine receptor. When you pull the stopper out of the ryanodine receptor, calcium comes pouring out into the cytoplasm. This is the beginning of a process known as excitation, contraction, coupling, frequently known as EC coupling. So the EC coupling then is where we've now released the calcium from the internal stores. The electrical event is the arrival of the action potential at the narrow gap between the T-tubule membrane and the sarcoplasmic reticulum membrane. That electrical event, because it's a positive charge, removes the blockade of the ryanodine receptor, and it removes that blockade of the ryanodine receptor by getting the dihydropyridine receptor to move the cytoplasmic region, which it had previously had stuck down the throat of the ryanodine receptor. That allows calcium to be released. So any questions to there? Yeah?
Just to make sure, is it sodium ions that are doing this, or is it more like an electromagnetic field that's propagating?
It's the electromagnetic field. It's the positive charge in the electromagnetic field. In this narrow gap, you'll have a lot of electromagnetic field because you have a lot of sodium, and it's a very, very narrow gap. So you get lots of charge, and the electromagnetic field and the charge moves a portion of the dihydropyridine receptor known as a 2,3 loop. It's a cytoplasmic loop, and that moves that 2,3 loop, moves it out of the opening in the ryanodine receptor, and calcium comes pouring out. The next step is now going to be use calcium to cause muscle contraction. So that's the contraction part, EC. That's the excitation, contraction, coupling. The excitation part is all electrical. The contraction part is going to be more mechanical using calcium. Okay. Anything else? Well, let's take a break. And we'll come back and we'll set up the concept of the cytoskeleton and talk about how the calcium is going to interact with the cytoskeleton to cause muscle movement. dimension. And that is this idea of acetylcholinesterase. Now you remember that what we emphasized in the central nervous system was the role of the glial cells in disposing of excess neurotransmitter. But we have a different arrangement here at the neuromuscular synapse. At the neuromuscular synapse, where here in red we have the postsynaptic membrane, and here in blue we have the presynaptic cell where the synaptic vesicles fused the plasma membrane and released the neurotransmitter. And of course we emphasize the idea that the neurotransmitter's role was to bind to the nicotinic acetylcholine receptors. But it left open the question of what happens to that neurotransmitter. If we have a really active lower motor neuron releasing lots and lots of neurotransmitter, I think you can see we're going to run into trouble pretty quickly in that we're going to saturate all these receptors. Turns out the first thing that happens is they'll all open, then they'll go into an activated state and they're not openable. But the point I want to make here is that in between the plasma membrane of the axon terminal and the plasma membrane of the muscle fiber, there is an extracellular environment. There's actually a basal lamina. And if we look carefully within that basal lamina, let me put it in green. If we look carefully within the basal lamina, we see that there are these proteins. They're extracellular proteins. And these extracellular proteins are said to be acetylcholinesterase. I won't get into the chemistry, but it turns out that acetyl, is covalently bound to choline by an ester bond. So if you have an esterase, we can break the ester bond. And when we break the ester bond, we convert acetylcholine into acetate and choline. Neither acetate nor choline is capable of binding to the receptor and opening it. So these are inert. So rather than what we saw in the central nervous system, we have these special cell types to take up and basically hoover out the extra neurotransmitter. Here we use an enzyme. We use this enzyme acetylcholinesterase so that what will happen here, it's a really interesting situation where the acetylcholine is released by the vesicle. The first thing that acetylcholine binds to is the esterase. But there are not enough esterase molecules here to accommodate all the acetylcholine, so there's excess acetylcholine. It spills over through this barbed wire meshwork and finds its way to the receptor, where it binds the receptor and opens it. Now, the esterase is a very fast molecule, so what's going to happen is the esterase is at first going to be saturated by the acetylcholine. But then, as it rapidly converts acetylcholine to acetate and choline, it now will leisurely sit there and then pick off individual acetylcholine molecules as they come off the receptor. Turns out that for this receptor to open requires 2 molecules of neurotransmitter. We can't reopen that receptor. After it's been opened, it will go into something called the inactivated state. It's effectively closed. We can't reopen this acetylcholine receptor until both acetylcholine molecules have come off. So as long as one of the 2 is occupied, it's in an inactivated state. And we use this, we commonly use this in surgeries, where if you're doing surgeries and you don't want the person to move, that you'll intubate them so that you're now in control of their breathing, and then you'll apply something known as succinylcholine. And succinylcholine is known as a depolarizing blocker. And what that means simply is the succinylcholine binds to the acetylcholine receptor. It opens it. but it doesn't keep it open very long. It goes into a closed state. But the succinylcholine can't be broken down by the cholinesterase. So there's succinylcholine here all the time, at one point or another, bound to one or the other of the receptors. So these remain closed. So your muscle remains relaxed. So, but all of that is a detail to say that the way we get rid of the neurotransmitter at the neuromuscular synapse is to block the acetylcholinesterase. And that's fundamentally how nerve agents work. Nerve agents like VX and Novichok, both of those will bind to the cholinesterase and make it inactive, permanently inactivated, taking it out of the system. So you no longer have any acetylcholinesterase. And what will then happen is you first get a contraction of the muscle, and then after that, these muscles will not contract because so much acetylcholine in here, because we lack the esterase, we've poisoned it, that these receptors now are all in the inactivated state, and the muscle is relaxed. You can't open, you can't activate it. And you'll suffocate because that happens to your diaphragm. There are also effects of nerve agents on the autonomic nervous system, but that's its fundamental effect on the neuromuscular synapse. But that's what I skated right by because I was eager to get to the contraction story. Any questions on that? So again, remember, this is the first event: release of neurotransmitter, generate an EPSP, and then that generates an action potential. And now what we want to do next is we want to talk about the role of the cytoskeleton. In muscle contraction. Now, when we look at the elements here, when we look at the players in the story, the first player we're going to talk about is something known as the actin thin filament. The actin thin filament has three components. First, it contains filamentous actin. But bound to that filamentous actin are two other players. One is called tropomyosin, and the other is called troponin. Turns out troponin is a trimer. There are three proteins in it. There's troponin C, troponin H, and troponin I. Don't worry about that, but know that the troponin, particularly troponin C, binds calcium. But all of these characters, the actin filament, the tropomyosin, troponin, all part of the actin thin filament. The next player here is going to be something known as the myosin thick filament. Now, myosin thick filament is a bundle of myosin molecules. And typically what you'll see in this bundle of myosin molecules is that their tails are all bundled together, but their heads are sticking out. And remind you again, when we talk about myosin, we see that it usually comes as a dimer, with those two tails, and then the two heads. So this is a myosin. But these myosins will be bundled together, and we'll see how they're going to be bundled together later. But know that the myosins are bundled into a thick filament. And the third player is going to be something called alpha actinin. Alpha actinin is an actin binding protein. It's an actin binding protein it's an actin binding protein that forms the structures known as Z-lines. So we're going to assemble this thing. We're going to assemble it from the actin-thin filament, the myosin-thick filament, and the alpha-actinin Z-lines. And I'm going to draw one of these muscles up here just like we had earlier. But now, rather than emphasizing the membrane system, what we want to be talking about now is the cytoskeleton. And these elements, the thin filaments and the thick filaments and the alpha-actinin, the Z-lines, are not distributed randomly in here. There's a very regular and repeating pattern. And it's such a regular and repeating pattern that it gives rise to skeletal muscle as being said to be striated. It's striped. What I'm going to lay out here is something known as the sarcomere. The sarcomere is the cytoskeleton laid out in repeating units. And what we're going to see is that these repeating units are bounded on their ends by these structures I'm going to put in black. Now, believe me, this is not to scale. A typical muscle fiber, even a small one, may be like a millimeter long. And each one of these sarcomeres, which are bounded by the Z-lines, each one of these is about 1.5 to most 2 micrometers. Some of them are as small as a micrometer, and I like to use the micrometer number because it's easy to work with. So those Z-lines, as we said down here, are actin binding proteins. So if we look at the structure, we'll see that the Z-lines have attached to them these actin-thin filaments. So in blue, we have the actin-thin filaments. And the actin-thin filaments are attached to the Z-lines. And then between the actin-thin filaments are these structures here. These are the thick filaments. These are the myosin-thick filaments. They have their tails pointing inward at the center, and the head's pointing outward. So if you look at this in cross-section, what you see is a myosin-thick filament with its head sticking out. 1, 2, 3, 4, 5. Should be a 6th one. And then around that actin-thin filament, around that myosin-thick filament will be 6 of these actin-thin filaments. What I'm attempting to show you here is that these myosin heads are arranged in such a way that the myosin heads are trying to reach up and contact the actin-thin filaments. So each of these myosin filaments will be surrounded by 6 actin-thin filaments. And the center of this structure is going to be the tails of the myosin. And the outer side is going to contain 6 of these. I only put 5 in here. So the myosins now are trying to reach up and contact that actin-thin filament. Let's now look at the actin-thin filament structure and see how it's organized. So what I'm going to do is go back to the upper panel and describe the structure of the actin-thin filaments. Remember, we said the actin-thin filaments contain 3 players. They contain the actin filament, they contain the tropomyosin, and they contain the troponin C, or the troponin complex. I'm going to do my best to draw one of these, and I'm going to show each one of these circles being a G-actin. So this is meant to be globular actin. We see that they're lined up end to end, but they're also offset a little bit. And this is filamentous. Actin. Put a few more in. Then in green, I'm going to now put the next molecule. And this green molecule is going to be something called tropomyosin. Tropomyosin is a ribbon-like molecule. And the way I typically draw, the way we typically think of it, is that it sits as a flat ribbon on the thin filament. Put another one out here. So this is intended to be tropomyosin. And I'll put a couple more actin monomers underneath it. Then the third character in the thin filament is going to be this structure I'm going to put out at the end of the tropomyosin. In black, this structure is meant to represent the troponin complex. Now, it's true there are 3 proteins in the troponin complex. The one I want you to know about is troponin C. The C binds for calcium. So the calcium, which had been released from the sarcoplasmic reticulum, is now out in the cytoplasm. And that calcium now is going to bind to the troponin C. When we bind the calcium to the troponin C, that's an enormous change in the charge on that structure. And by changing the distribution of charge at the interface between the troponin C and the tropomyosin, what will happen is that this tropomyosin molecule moves. Actually, it looks like the troponin C is holding it in place. And when calcium binds to the troponin C, it now moves. And when it moves in such a way that it's no longer in this flat arrangement, but rather, let me draw it out here. I'll just draw one of them. Rather, it's effectively turned on edge. Now, if we go back to this image, we find that each one of these actin monomers, each one of these contains a binding site on it. And I'll put this as a red dot. And this red dot is the myosin binding site on the actin thin filament. So if we go back up to this image, we can imagine that these are the myosin heads reaching up, trying to contact the actin thin filament. So if my arm is the actin thin filament and my fist of my right hand is the myosin head, it's trying to reach up and contact that actin thin filament. But the problem is that there's a molecule of tropomyosin in the way, so it can't make that contact. So another way of thinking about this, and the way I drew it on this panel, is that these myosin binding sites in the actin are covered over, they're occluded, they're blocked by the tropomyosin. So you can imagine the myosin head down here trying to reach up and contact it, but it can't. But now, when calcium is released from the sarcoplasmic reticulum, the calcium is released from the sarcoplasmic reticulum, it binds to the troponin C. It folds the tropomyosin down into the groove between the actins, and now we have an open site here to which the myosin head can bind. So you now can imagine the myosin head being my fist. It's going to snag on to that binding site. If we look at it in a slightly different fashion, let's sort of show it to the side. Put it down here. And if we look at it here, here's the myosin binding site, right? We've opened that up. Now the myosin head can reach up and contact that binding site on the actin. Now, you all okay with that? So we have yes?
What exactly happens to the tropomyosin shape after calcium binds?
So what happens to the tropomyosin after it binds is it moves. It folds. How to explain this? So the myosin binding site in the actin is covered over by the tropomyosin. So the way the site's opened is that the tropomyosin has a favored position on the actin thin filament. And the favored position, when it's not interacting with the troponin C, is to be tucked down into the fold between these actins. So that's why I tried to show it here is on edge. So it's flat here. Then it moves down into the fold, and when it moves down into the fold, you only have to move a few fractions of a nanometer, and you've now opened up the site here so the myosin head can reach up and contact it. So the first step is to expose the binding sites. And then we can now think about what's going on with the myosin as it contacts the actin thin filament. Well, I'll remind you from something we talked about early on, that each one of these myosin heads has a binding pocket, an ATP binding pocket. So it will bind ATP at this site. And another important concept here was that the myosin head is an ATPase. The myosin head is a really, really efficient ATPase. So almost immediately after the ATP binds to that binding pocket, the ATP is converted to ADP and inorganic phosphate. Adenosine diphosphate plus inorganic phosphate. And it can sit for hours. That's stable inside this binding pocket. When it becomes unstable is when the myosin head reaches up and contacts the actin thin filament. The instability here is such that the ADP and PI leave. The loss of ADP plus PI initiates the power strip. What we mean by that is that when this myosin head is attached to the actin thin filament, we have these sort of attached to one another, and the ADP and PI leave, that loss of all this charge with the phosphate and the ADP from the binding site causes this myosin head to move. It effectively bends over. It goes from an upright position to being bent over. When it's bent over, there's a mechanical movement of this head. Now, I'll extend this out as just a line. But this is going to be attached to the Z lines. So let's go back up to the upper panel. So we've released the calcium from the internal storage compartment. The calcium's poured out through the Randy receptors. It's bound to the troponin C. It's moved to the tropomyosins, and these myosins now are available to reach up and contact the actin thin filament. What we talked about above is that these myosin heads, as they're reaching, contain ADP and PI. But as soon as they contact that actin thin filament, the ADP and PI leave, and that's the power stroke, and it will bend over. Well, you can see here that these are going to ratchet in this way. They're going to walk along the actin thin filaments like this. They're going to draw these Z lines together. You're doing this, and these Z lines, these sarcomeres, are set up in series. So if you shorten this by 20% and shorten this by 20% and shorten this by 20%, you're going to now lead to the shortening of the entire muscle by 20%. So even though this sarcomere shortens from, say, 1.0 micrometers to 0.8 micrometers, other way, even though we've only changed this bit by 0.2 of a micrometer, because everybody's laid out in series, that leads to a 20% reduction in the length of the muscle. That's how we shorten the muscle. So there's a question in the back.
Yeah, so after the myosin head like flops over, how does it reset that?
That's the next point, right? So it's a really good question, because now what I've told you is that the myosin head is locked onto the actin thin filament. It's locked on, and now we have an empty site here. And your question is, how do we change that arrangement? The next step in the story is that it requires another molecule of ATP. Remember, we have a tremendous number of molecules of ATP sitting in the cytoplasm. And so this ATP now can move into this site, binding ATP to a myosin head attached to an actin causes the myosin head to detach and recock to its original position. So in other words, the question that was asked is, once we have attached this myosin head to the actin and it's pulled on the actin thin, and it's pulled on the Z line, how do we have it let go? The way it lets go is we bind actin. The way we have it let go is it binds ATP. The myosin head now returns to its upright position, and it's now strategically placed to contact the next actin monomer and undergo the process again. So as long as two conditions are met, as long as there's calcium out there, and as long as there's available ATP, we'll continue to go through this cycle. We'll bind the calcium, we'll move the tropomyosin, we'll then use the myosin head to bind the actin thin filament and undergo a ratcheting. But for us to reset that system, we need ATP and to recock it. You had a question again.
Yeah, so then where does that ATP come from?
That ATP is coming from the mitochondria. It depends a little bit on which muscle you're talking about. I don't want to get into fast twitch and slow twitch muscles, but in fast twitch muscles, there aren't many mitochondria, and much of that ATP is coming from glycolysis. In slow twitch muscles, which ones you're active for long periods of time, are predominant long distance runners and bicyclists, for example, those ATPs are coming predominantly from mitochondria. And we'll see some muscle fibers have way more mitochondria than others. But in the ones that have few mitochondria, that's glycolysis. They don't last very long. I mean, they run out of ATP pretty fast. Whereas the ones that are generating ATP from oxidative phosphorylation of the mitochondria, they can remain active for a long time. That's where the ATP is coming from. And it's throughout the cytoplasm. If things are healthy, you'll have at least a millimolar concentration of ATP in there. They're everywhere. Yeah, do you have a question? No. Okay. So that opens the question, how do we stop this whole thing? We've released the calcium in the internal stores. Calcium's bound to the troponin C. It's moved the tropomyosin, and we've gone through this process. Well, we go all the way back now to the role of the sarcoplasmic reticulum. Now, up to now, our emphasis has been on the ryanodine receptors, which are allowing calcium to come out of the sarcoplasmic reticulum. But there are another set of proteins on the SR. I'll put these in red. And these are called calcium ATPases. These are calcium pumps. They use ATP to take calcium, which is in the cytoplasm. Remember, we dumped a lot of calcium from the sarcoplasmic reticulum out into the cytoplasm, and these pumps now are busily moving calcium back into the SR. Now, as long as the action potentials, which are initiated at the neuromuscular synapse, as long as those are arriving at the TSR junction, and those calcium channels remain open, we're pouring calcium into the cytoplasm. Calcium is binding to the troponin C, moving the tropomyosin, allowing the myosin heads to contact the actin thin filaments and pulling those Z lines together, as long as we have calcium. But when those action potentials stop arriving at the neuromuscular synapse and no longer generate an EPSP through opening the ligand-gated ionotropic channels, the nicotinic acetylcholine receptors, no longer setting up action potentials, no longer delivering those action potentials to the TSR junction, since we no longer have that charge of the sodium in between the T-tubule membrane and the SR membrane, that plug moves back into the bottle. We stop the release of calcium. So when the action potentials stop, the calcium release from the internal stores stop. And now these guys take over. Calcium pumps take over. And they start pulling the calcium out of the cytoplasm and putting it back into the SR. So as the calcium is being pulled out of the cytoplasm and put back into the SR, the cytoplasmic concentrations of calcium are dropping really fast. The troponin C has an ability to bind calcium, but it doesn't bind it very tightly. So as long as there's plenty of calcium around, it will have that, the troponin C will have the calcium bound to it. But as those calcium levels drop, now the calcium starts to come off. The law of mass action says the calcium is going to be coming off these troponin C molecules as the calcium concentrations drop. When the calcium comes off of the troponin C, it no longer tells the tropomyosin to be folded down into the groove, but instead flops back up over the actin binding sites, or the myosin binding sites on the actin. So now that's the way we end contraction. The way we end the contraction is we pump the calcium back into the sarcoplasmic reticulum. Let me finish that story with kind of a grisly tale. And the grisly tale is something we all know. After an animal or a person dies, it becomes stiff. And that stiffness is called rigor, or rigor mortis. This is the stiffness of death. Why that stiffness of death occurs is because of the story I just told you. And that is, after death, we no longer are delivering oxygen to the mitochondria. We're no longer making ATP. So as death comes on, now ATP production begins to drop. So inside these muscles, we no longer have very much ATP. That's a double whammy, because we no longer have ATP to power the pumps. So calcium remains in the cytoplasm. So the calcium levels in the cytoplasm start to go up. Because the calcium levels are going up, now they've bound to the troponin C, moved the tropomyosin, and the myosin head has contacted the actin thin filament. But remember, our ATP levels are low. So now we don't have any ATP to bind to the myosin head and let it come undone and recock. So what the end result is the myosin head is permanently attached to that actin thin filament, and that's rigor mortis. And pathologists, forensic pathologists, know all about this. They can estimate the time of death by estimating the stiffness of the body, knowing something about the local temperatures and how long the body's been there. Now, after a period, I think it's like 72 hours or so, you begin to have a bunch of enzymes that take over, that come out of the lysosomes, and they start breaking the whole thing down, and the body goes limp again. But if you understand that grisly tale of why the body becomes stiff after death, you can puzzle your way through what's going on with muscle contraction and, more importantly, muscle relaxation. The final point I want to make on this is that this is something that's going to come up over and over and over again. We have the ability to actively shorten those sarcomeres, contract the muscle. What we don't have the ability to do within that muscle is to bring it back to its original position. I think you can see that when this muscle shortens, we're going to shorten the Z-lines. But now the question is, how do we reset those Z-line lengths? Let's say we've reduced the Z-line length, if we have reduced the distance between the Z-lines, if we've reduced the sarcomere length from 1 micrometer to 0.8 micrometers, how do we reset it to 1? That requires antagonistic muscles. So that all of your muscles are set up in antagonistic pairs. The easiest one to think about is your biceps and triceps. When I activate my biceps, I pull my hand up to my shoulder. Now, I can stop activating that muscle, but in order to reset its length, I have to activate the triceps, contract the triceps, and extend my arm to reset the length of the sarcomeres in my biceps. It's true for all our muscles. I think of it as like pushing on a string, that once you've contracted it, something else has to act to restretch it. And those are the antagonistic muscles. And we'll talk about this any number of times. We talk about respiration and talk about breathing, and we talk about cardiovascular system. When the muscle contracts, how to return it to its original length. Skeletal muscle is easy. Skeletal muscle, you just have the antagonistic muscles, like we said with the hamstrings and the quadriceps. But it will get more complicated when we start thinking about the heart, and a little bit more complicated when we start thinking about the lungs. But that pretty much brings us to the end of the second section, and Monday's exam will cover nerve and muscle. Sorry I didn't get a smooth muscle. Sometimes I make it, and sometimes I don't. And Abby's happy because she doesn't have to explain it now. Well, thanks, and we'll see you Monday for an exam.